6:32. We're going to start. Before we start, I'll just introduce myself first. My name is Thaw. It's right in my screen name. Thaw. I'm a third year. I took Bio30 2 years ago with Dr. Wilkinson. It's been a while since.
My name is Fatima, everybody. I took Bio30 with Dr. Wilkinson last fall, so it's been a year since I took it. I'm also a biology major, and my concentration is systems physiology. Hi, Karen. I just saw the message. So it's my second year here at SJSU, but Thaw is third year.
Yeah, I'm a third year.
Yeah. He has more experience.
Oh, it's my second semester working with Peer Connection. And this is my first time doing virtual essay sessions. So if we're like struggling, please be understanding because this is our first time.
Also, if you have any feedback, you can tell us. You can email us. That's probably the best way you can tell us. I'll just send in our emails in the chat right now if you don't know what they are.
Right. So transcription, translation, and Lac Operon are the three things that we're going to go over today. So first question of the day, can you guys describe what transcription and translation is to us right now in the chat box, right?
Just like 1 or 2 sentences, what it is, kind of like why do we need it, why it's done, and what is the point? So you can just send like 1 or 2 sentences in the chat box right now. Going from genes, DNA, to RNA, to proteins. That's actually very good. That's a very good summarization. That's exactly what it is.
And Neha said transcription is when DNA is pulled apart and formed into RNA.
That's true.
And Karen said central dogma.
Very excitedly. That's also true. It is the central dogma.
Okay, so we got some answers, so we'll just move on to describing what transcription is. Yeah. So like Karen said, transcription is going from DNA to RNA, DNA to RNA, but starting from RNA to protein is called translation. So we'll talk about translation a little bit later, but I'll start with transcription. So transcription, right here, you see a DNA sequence, right? So what's going to happen is that it's going to be split into 2, and then there's a protein going to come in called RNA polymerase. It's going to attach to a starting codon on the DNA strand, and then it's going to start transcribing to an RNA strand. And there you have your mRNA. This transcription happens in the nucleus. And where do you guys think that mRNA is going to go next?
Also, I just want to clear up. So this DNA, when it opens up because we're going to start transcription. And what signals transcription is when the enzyme RNA polymerase reads the first sequence, it's called the promoter region, but it's what gets it started. So when RNA polymerase reads the first few bases of the DNA, it knows to start transcribing. So then it starts transcribing and then synthesizing an mRNA strand.
So we have 2 answers that say cytoplasm, and you guys are right. Yeah, mRNA is going into the cytoplasm.
Oh, also one thing. You see when both the strands open up, only one of them is used as a template, right? The other one isn't used. Does anybody know why? Why only one of them? Why isn't the coding strand, which is the one on top, why isn't that one copied? So the coding strand, it has the genes that we actually need, right? It has the genes that code for proteins. So the reason why we don't copy the coding strand is because synthesis of mRNA is complementary, just like DNA replication. So if there's A on top, the mRNA is going to synthesize a U, not a T, because there's no T in RNA. So it's going to synthesize a U. So if you want the sequence that's on the top and you copy the top one, you're not going to get that sequence. So if you want the sequence that's on top, you need to copy the other strand, because it's complementary. you want us to go over in more detail, just tell us.
I did not get the last part. So the last part is about the coding strand.
Okay, so the coding strand, as you can see in this picture, can you point at it?
That's like,
Yeah.
okay, yeah.
So as you can see, the top strand right here, it's labeled as the coding strand. So what the coding strand is that it has the genes. So every code on this strand codes for a gene that codes for a protein. So when it's translated, these are the genes that we want to translate into proteins. So they're on the top strand. And the bottom strand is just the bases that are complementary to those. They're not the genes. So if we want the genes on our mRNA strand, because we want to translate the genes, right? So we have to copy the bottom strand so we can get the complementary base pairing and then end up with the top strand, but in mRNA form.
Okay, before I jump to the translation part, there's another process called post-transcription modification splicing. So basically, what you guys need to know here is that so your mRNA has this thing, this parts called exon and introns. So what's post-transcription modification? You're just slicing off all the introns because you don't need it in your translation process.
Wait, I have a question.
Yeah.
So why don't we need introns?
Wait, it's been a while. Okay. Is it because introns are just like extra like genetic sequences that you don't like when you don't really like need those to make the protein?
Yeah, they don't code for anything. It's basically just like a buffer region. They don't code for proteins, so we don't need to translate them. They code for something else, but they don't code for proteins. So we don't need to take them to translation, waste so much resource trying to find a protein when it doesn't even have one. So we just remove the introns because they're non-coding regions, and the exons are the coding regions. So the exons are the places where there's codes for protein. So we're interested in the exons. So we remove the introns, and the process is called splicing. It sounds like slicing, right? You just slice it up and then throw away the stuff that you don't need. And then you have your mRNA strand ready to be translated. So here we have the top part, as you can see.
I'll move my
Yeah, so this is the top part of the ribosome. This is one ribosome, by the way, but it has two parts. There's a large subunit and then there's a small subunit. And as you can see, our mRNA strand is on the small subunit. So it actually goes and binds to the small subunit. It makes a bond with it. So if they have a bond in between them, do you think they're gonna move? Do you think they're gonna move because they're joined to each other, right? So they're not going to move, and the only unbonded thing we have here is the large subunit. So what's going to happen is that the large subunit is going to move along on the mRNA, but the small subunit and the mRNA is going to stay in place. And you see here, there are these sites on the ribosome, the E, P, and A sites. So A site is, you can remember it as acceptor. So that's where the first tRNA comes in. And in the corner up there, you can see what tRNAs look like. So they have an amino acid on the top, and then they have an anticodon on the bottom. So the first tRNA comes, it comes onto the A site, it's accepted by the ribosome. Where does the larger subunit move to? It moves along the mRNA. So the mRNA stays in place, and the large subunit just like moves on.
Yeah, so the anticodon, so you know how the tRNA has that bottom part where the anticodon loops is? So basically there are genetic codes that are the opposite letters of whatever is on the mRNA strand. That's how the tRNA move along the mRNA strand.
Yeah, so they come on with their anticodon. So if the specific part on the mRNA is AUG, then the tRNA is going to have what's complementary to it. So it's going to have AUG. It's going to have UAC. So it's going to have UAC. So it's going to have whatever is complementary, and then they're going to like bind together. They're going to like pair up and stuff. And then that's how you know that this tRNA matches this specific amino acid, right? This specific amino acid is what we need in this situation. And the first one, always remember, is AUG. That's always the first amino acid, no matter what protein it is. The first amino acid is always AUG. And that's also called the start codon because it's always first. So then the yes, that's why it's called anticodon. So on the mRNA we have the codon, and then on the tRNA we have the anticodon. So it just means that they're complementary to each other. Okay, so AUG, as I was saying, is the start codon. It does code for an amino acid. It's called methionine or something like that, but you don't need to know that. But just know that it's always the first amino acid, no matter what kind of protein it is, because AUG is the start codon. So it's the start codon. It also has an amino acid, and then once the amino acid and once the tRNA has already binded at the acceptor site, the large subunit moves along, and then the first amino acid, which was in the A site, is now at the P site because the large subunit just moved one space up, right? So the first one is now at the P site, and then a new one comes on the acceptor site, and then its anticodon and the codon pair up with whatever is on the mRNA, and then the amino acids on top, they actually form a bond. The bond is called a peptide bond. It's kind of a covalent bond, and the word peptide is just specific to the bond between amino acids. So it's used for proteins. So they have a covalent bond that forms in between the amino acids on top. So, okay. Oh, I have to skip for this. So as we said, this one moves along. I'm just gonna have to group this again. I'm sorry. Okay, so this one moves along, and then the first tRNA is now at the P site, and then we have a new tRNA that comes here, and then they form a bond on top. So I'm just gonna like draw. So this is a bond between them.
Neha asked if anticodon binds with mRNA and the amino acids bond together. So
anticodon doesn't bind. It matches up to confirm that this is the right amino acid. So they don't actually have a bond between them, but they do match up because the tRNA, you know, it's gonna you're seeing a bit, but it's gonna leave soon. So they don't actually form a bond, but the matching happens to confirm that this is the amino acid that goes in this sequence right now. And then the ribosome moves one up again once they formed a bond. So now the first one that we started with is at the E site, and the E site actually, you can think of it as the exit site because this one is going to leave now. So it goes away to find another amino acid that matches with its anticodon, and then, oh yeah, its amino acid stays. No. Okay, I'll draw on it. So I'm gonna. So imagine that the amino acid stayed, okay? The tRNA left, but the amino acid stayed. And then the A site is empty again. So we are going to have another tRNA come in and then match with the mRNA sequence, whatever is next. And then again, they form a bond between them, just like the other ones did. And then what you end up with in the end is just a long chain of amino acids bonded to each other. This just keeps happening. The tRNAs come, they match up with their codes, and then they leave, but they leave their amino acid there because they're bonded. So you end up with a long chain of amino acids, which is called a polypeptide because there are a lot of them, and they're bonded to each other with a peptide bond. So this is what you end up with, and this is your protein. It's gonna there's some other steps that it has to take before it's actually ready to function, but essentially this is your protein and this is the end of translation. So if you have any questions, send them in right now.
I do have a question though.
Yeah.
All right, so how do you know when translation stops? So, like, what does it need to happen
Oh,
for
yeah.
translation to stop?
So for the last one, whenever the last tRNA comes in, so I'm gonna move this one up again. So now this is our last tRNA, but there's one thing different about it, that it doesn't have an amino acid. It only has an anticodon, which is a stop codon. So it doesn't have an amino acid. It's a stop codon. Whenever it matches up with the last sequence on the mRNA, it knows that translation has stopped, and because it doesn't have any more amino acids to bind in the chain. So when this happens, when the mRNA sequence reads that the anticodon is a stop codon, which are also specific, by the way, just like the start codon, there's like 2 or 3 stop codons as well. But the key thing to remember is that they don't have an amino acid. It's a specific code, which means that it's time to stop. So when that happens, the mRNA dissociates whatever bond that it had with the ribosome. It dissociates and then it goes back to the nucleus to be reused. And what we end up with is our chain of amino acids, which is ready to be folded and become a protein.
Karen asked when does folding occur, like right away before the stop codon, or like after the stop codon?
After the stop codon. So when the stop codon is read, that's when translation stops and the mRNA detaches, and so does the amino acid chain. It also detaches, and then it goes to whatever organ it needs to go to start folding up.
So Priya asked, what might be a little late, but what does the small subunit do?
So the small subunit does nothing. It just holds the mRNA in place. It does nothing else. The whole job is done by the large subunit. So when the mRNA comes in, essentially before starting translation, it binds with the small subunit so it can stay in place. And then the large subunit can start moving without everything just floating around. So the only thing that the small subunit does is it holds the mRNA in place.
So think of it as like something that is like holding mRNA back. It's just like grabbing onto it.
Yeah, because things around it are moving, right? So, and the cytoplasm, it's fluid. Things are always moving around. So you need to hold it in place because you need to do this right. You can't mess up, or you're gonna mess up your protein, and it's gonna mess up the function. It's just gonna be a big mess up. So it's kind of, you can think of it, it's kind of like a protective effect. You just need to hold it in place so you can do the job correctly. So that's it for translation.
Okay, if you guys have any more question, just send it to the chat and we'll answer it toward the end of the session, if that is fine with you guys. We in the essence of time, we do have to move on. So the next thing we're going to talk, oh wait, is there screen?
Oh yeah.
The next thing we are going to talk about is Lac Operon. So something that I usually ask my students for my sessions are to like know how much they know about the Lac Operon. Like, so if you guys want to answer it, please do in the chat box and we'll read your answers. So my question is, what is a Lac Operon?
Yeah, why do you think you need to study it? How does this apply to whatever you studied?
Yeah.
Because I know your professor went over this, but I know a lot of people were confused as to how this relates with your topic. And a lot of people aren't clear as to what Lac Operon actually is an example of. Okay, somebody says it's an example of gene regulation. Somebody said might transport and metabolize lactose. So all of that is correct. It's definitely to do with lactose, and there is transport and metabolism of lactose involved in it, and it is gene regulation. But more specifically, it's an example of transcription in E. coli.
All right. So
Yeah, so you just want to define those.
So, okay, in the Lac Operon, there are two gene sequences that code for two proteins, okay? So first one that I'm showing right now is LAC-B. And LAC-B is a gene that encodes for beta-galactosidase. Galactosidase, okay. Beta-galactosidase. And what beta-galactosidase is that is an enzyme, and it breaks down lactose into glucose and galactose. And there's also, oh yeah, so there's a box for P. There's also LAC-P. LAC-P is just another protein. It codes for a permease. And what that protein do is that it transports lactose into the cell.
So it can be used.
It can be used. It can be used to break down
into glucose and galactose.
Yes. And you also have LAC-I, and LAC-I is the inhibitor of LAC-B and LAC-P transcription. So as long as LAC-I is there, transcription is not going to start. So we represent it as a really big box. LAC-O is the operator region. So on your DNA strand, as we described earlier in the session, there's going to be an operator region in your DNA strands for the RNA polymerase to bind to and start the transcription process. Yeah. Okay, so in the diagram you see an RNA polymerase waiting for transcription to start, but because LAC-I is there, RNA polymerase cannot proceed to code to synthesize the mRNA strand that needed to make the protein.
Yeah, so basically the operator region is the pathway between RNA polymerase and the mRNA strand that it needs to go and transcribe, and LAC-I is standing there blocking the way. So why is LAC-I blocking the way? Why is LAC-I inhibiting? Why don't we want this to happen?
Okay, we don't want this to happen. We want LAC-I to be blocking when there is no lactose. Because if you make the proteins when there is no lactose present, it's just a waste of energy for the cell. So you don't want that in your body. Like you don't want to waste energy doing unnecessary stuff, right? So you only make these two proteins when lactose is present in the cell.
Yeah, and that's because when there is no lactose, that means that glucose is there and it's being used. So you don't really want to waste your energy and your time and your resources trying to break lactose into glucose and galactose when you already have glucose. So you can just go ahead and use that. So that's why LAC-I is always inhibiting the process unless if lactose is there. And that is because lactose is actually an inhibitor for LAC-I. So lactose is when it's there, it stops LAC-I from doing its job, and its job is to block the way, right? So when lactose is there, LAC-I just falls off, and you can go on the next slide and I show that. Yeah, so that green dot over there is the lactose.
Okay, so in this case, lactose is the inducer and LAC-I is a repressor. And what lactose is basically doing is that it's inducing LAC-I to go off of the strand so that RNA polymerase can start transcribing the DNA sequence. There you see. So LAC-I goes off of the strand. RNA polymerase starts transcribing. And you get your mRNA strand.
This is to show what LAC-O is.
Okay. So, do you have the table by any chance?
Yeah, I'm doing it right now.
Okay. So I'm gonna stop. We're gonna switch to Fatima's screen again.
If you can just keep showing that slide, I just want to add something.
Okay.
So I wanted to add that the operator region, or the LAC-O, the gene that codes for the operator region, if Lac O, you'll see in a minute what I mean, but if Lac O isn't present, then that means that this pathway, this pathway, you can see the squiggly line on top of it, right? That means that it's broken. So RNA polymerase can't go through because the path is broken because its gene is mutated. So when when Lac O isn't there or it's mutated, this process can't happen. You can transcribe lactose whether LAC-I is there or not, whether lactose is there or not. Lac-O, if it's faulty, if it's mutated, you can't do this process. Okay. So this is a table that shows the different types of conditions in which we could use this. So here in this column, you can see all these genes. So you can see LAC-I, which if you remember is the inhibitor. So when lactose is there, on this side, by the way, is beta-galactosidase transcription. So this is just the rate of transcription. So when it's 100, that means it's being transcribed. When it's 1, it means it's barely being transcribed. So you can see when LAC-I is there and lactose is there, there is transcription. Because if you remember, lactose is an inhibitor for LAC-I, so it actually makes it fall off the pathway, allowing RNA polymerase to start transcribing beta-galactosidase. So that's why we have a lot of transcription here. But when there isn't any lactose and there is LAC-I, and these plus and minus just mean there and not there. So when there is LAC-I but there is no lactose, we don't have any transcription, or we have very little transcription of beta-galactosidase, and it's not enough to go through with this process. And then if we don't have LAC-I and we have lactose, we're going to have a lot of transcription. Even if we don't have lactose, we're going to have a lot of transcription because there's nothing stopping it from happening anymore. So if you take LAC-I out, or if LAC-I doesn't do its job, you're just going to have a lot of transcription of beta-galactosidase. We're going to waste a lot of resources and time and energy trying to get glucose from lactose when glucose is already present. And then here, LAC-O, this negative just means either it's not there, or it's mutated, or it's faulty. So you can see when lactose is there and LAC-O isn't there, you barely have any transcription of beta-galactosidase because remember that pathway is broken, so your RNA polymerase can't reach the mRNA strand, the DNA strand to start transcribing. And then same here, it doesn't matter whether lactose is present or not. If your pathway is mutated, your RNA polymerase can't reach and start transcribing. And then same here, you have LAC-I, which is an inhibitor for this process. And you have lactose, but your LAC-O is mutated, so you don't have any transcription where you should, right? When your LAC-I is there and your lactose is there, you should have transcription. But you can see if your LAC-O is mutated, you don't have any transcription. And it's same thing, it doesn't matter whether lactose is there or not. If your LAC-O is mutated, you don't have any transcription.
I have a question.
Yeah.
So let's say you don't have a LAC-P.
Mm-hmm.
What do you think the number of proteins going to be if there is lactose present?
So LAC-P just codes for another enzyme. So I don't think that LAC-P would affect the transcription of beta-galactosidase, but what it would affect what happens next, because remember LAC-P permease actually transports the lactose into the cell, so this process can happen and we can start using glucose as energy. But if you don't have LAC-P, if you don't have permease to transport that lactose, you're basically just breaking up all this lactose, but you can't use it because there's nothing to transport it. So all of these genes are very important. And they're all on the same chromosome, so they're all like next to each other. So when they're transcribed, they're all transcribed together.
Neha asks how the ASM is going to be. From what I heard is that the ASM is going to be multiple choice, and you do need to know everything, but the main things that I will focus on would be the experiments at the beginning. So Griffith experiment, Avery experiment,
Hershey Chase.
Hershey Chase, and Meselson and Stahl experiment.
And that one's very important.
Yeah, so those experiments are very important to know. In addition to that, you also do need to know the three models of DNA. So semi-conservative, conservative, mosaic. Be able to explain like why DNA is a semi-conservative model and not the other two. Yeah. So.
So you don't have to know the experiments in detail, but what you should know is the graphs and the figures that your professor put in the PowerPoint, because often what happens with questions is that they give you that graph and then they ask you a question and you need to read the graph or you need to understand the graph, also with the understanding of the actual process that the graph shows. So it's not going to be like the answer is in the graph. The answer is in the graph, and then you need to apply the knowledge about the process that the graph is about. So if there's a graph about translation, it's going to ask you to find some value, and then it might ask you to explain why this value is like that. So for that, you would need to know how translation works.
Neha asked, "What's the last experiment? Do you mean Meselson and Stahl experiment?"
So that one was about DNA replication, what model it uses. And remember the three models were semi-conservative, conservative, and mosaic. So a question that Dr. Wilkinson likes to put in there is she's going to ask you, what would the band for mosaic look like? Or what would the band for semi-conservative look like? So if you read the experiment, you're going to know that they tried out all three and then they predicted what kind of bands they would see, if it was conservative or if it was semi-conservative or if it was mosaic. So there's a question sometimes where she asks you what kind of band it's going to show. So be sure to know that.
Okay, Riley asked, what was semi-conservative? Okay, first of all, do you want to answer those questions later, or do you want to move on to the recent experiment?
I think they want to ask the questions right now.
All right, so we'll just answer the questions for the remainder of the session then. Okay. Semi-conservative model.
So semi-conservative is a model of DNA replication, and it's actually the one that was, the one that was proved, right? And it is the one that we know that that's how DNA replicates. So do you want to explain how that works?
Okay, so semi-conservative model for DNA replication. So how I see it is that your DNA is double-stranded, right? So when that DNA replicates, it's going to create two new strands. So is there a way to visualize this?
I can share it with them and then draw it out.
Okay.
Yeah. So I'll just screen share with you guys and then draw what semi-conservative would look like. So let me do that. Okay.
All right. So semi-conservative. So you have your old DNA strands.
So let's draw them in blue.
Okay. So blue is your old DNA strand. Okay. So when they replicate one time, you're going to see two new strands. Can you use red for that one?
Yeah. So how this model works is that the two strands separate and then you make two new strands and then they pair up.
Yeah, it's going to look like the old strands are sandwiched in the new strand.
Yeah, so let me just draw this. So this strand goes here and then, so this strand, the blue one, it comes here. And then the new strand that's synthesized is this one. And then the same happens for the other strand. It goes here and there. And then it has a new strand created that's complementary to it. And then these strands are gonna form a double helix. So these are your two new strands. So how this process works is you have one strand of, you have one pair of DNA strands. They separate and then you make complementary strands for each of them and then they pair up. So this is semi-conservative, where you just have a little bit new and a little bit old. So that's why it's semi.
So my question to everyone here is, what would that DNA strands look like after the second replication?
Yeah, so remember in Meselson and Stahl, the first time they grew the first strand of DNA, the first pair of DNA in nitrogen 15, and then they changed it to nitrogen 14, and then they allowed them to replicate. So if I can just go to a new slide, and then I'll draw what that looks like.
Okay, that was a little choppy. Can you repeat the question? Yeah. So what I'm asking is that what would the DNA strands look like after the second replication
of the Meselson and Stahl experiment? So remember when they changed to nitrogen? So if you want me to draw that out for you, so the first strand of DNA, it was N15. So N is in the bases, right? Because the bases are nitrogenous bases. So the first strand of DNA was in N15, and then they changed the environment to N14. And then they let this old DNA replicate in N14. So the new strands, like if we have new strands created, they're going to have N14. They're going to look a little different because N14 is lighter than N15. So if you centrifuge it, which basically separates based on weight, so you're going to have N14 on the top and N15 on the bottom because N14 is lighter and N15 is heavier.
Someone asked to explain the mosaic model.
Okay, so let me just quickly go over the semi-conservative and then I'll explain the mosaic. So for this one, if they allow this DNA to replicate, and if this is, remember this is semi-conservative, so let me just write that here. Oops. So if you allow these DNA to replicate in the new environment of N14, we hypothesize that the two new strands that are going to pair up with the two original strands are going to have N14, right? Because that's the only nitrogen that's available to them in this environment because they changed it. So the new strand is going to have N14, and here as well, new strand is going to have N14. And then if we allow it to replicate one more time, so this one goes here, and then its new strand is also going to be blue, and then this red one goes here, and then its new strand is also going to be blue because nitrogen 14 is the only one that we have available. And then same here, this one's going to be blue, its new one is also going to be blue, and then this one, the old one is red, but the new one is going to be blue. So as you can see, there's a lot more blue than red in the third generation. So 1, 2, 3. So in the third generation, there's a lot more blue. So if you keep allowing them to keep replicating, at a certain point, there's just going to be like so much blue and very little red left. So if they saw this in their centrifuge, in their test tube, they would see, because each band that I'm going to draw in the test tube represents one pair of DNA, so both strands. So for the first one, because it was both N15, it's going to be on the top and it's going to be red because they're both N15. And then for the second replication, it's red and blue and it's half-half. So it's going to be a little lower. The N15, it should go on the bottom. Sorry.
N15 is heavier.
So the heavier one's going to be on the bottom. So N15 is going to be here. Both the strands have N15. And then in the next generation, there's red and blue, and it's half-half. So it's gonna be a little higher because it's a little lighter, but it still has some N15, so it's still gonna be dark. So it has both here. Why did that one just go away? Okay. So for the second replication, it's gonna have both red and green. And then for the third replication, because there's way more blue than there is red, what you're gonna expect to see is it's gonna be even higher because it's even lighter, because there's a lot more blue than red. So it's gonna be very high, and it's gonna be mostly blue, but it's still gonna have a little bit of red in it, very little bit. So this is what it would look like for semi-conservative for 3 replications. So do you want me to go over Mosaic now?
I explained it in the chat, but if you do want to, you can. But do you mind explaining why N15 is heavier than N14?
Yeah. So these numbers next to the N and 15 and 14, these are actually the mass numbers of the atoms of the nitrogen. So 15 is going to be heavier, right? Because 15 is bigger than 14. And how these two differ between each other is that N15 has an extra neutron, so that's how it has a little bit of extra weight, and it's heavier than N14. So we can look at Mosaic now. So for Mosaic, the strand is kind of a mix of both. So if we start in N15 first, the whole strand is going to be N15. But then when we allow it to replicate, because the Mosaic theory says that you just cut up bits of both strands and then just like make new ones and use some of the old ones. So what it's going to look like after one replication is a mix of both. So it's going to look something like this after the first replication, and you're going to have 2 of these because it replicated. Let me just draw 2 quickly. So that's one pair, and this is the other one. So remember, the blue is the N14 and the red is the N15. So this is after the first cycle. Oh, sorry, the second cycle. Oh, this is 1. This is 2. And then I'm going to draw 3 over here. So after the third cycle, we're going to have some more, well, a lot more of the N14, so a lot more of the blue, but we're still going to have some red because according to this theory, we still use like half of our old DNA and half of our new DNA. Oops. So it has a little bit of red, but mostly blue, mostly the new nitrogen. And then again, you're gonna have 2 pairs of them. So I'm not gonna draw the second one because I feel like it's a waste of time. They look exactly the same.
Someone asked in one of the sliders for lecture, there was a slide with different shades.
Well, the color doesn't matter. It's just to show that they're different.
I think he's talking about the microscope slides where you see like a dark band in the middle, and then you, as DNA continue to replicate, you see a darker band toward the left and a lighter band like on the right. So what it's saying is that you have more new DNA and the old DNA. That's why your the very left band is darker than the right band. So what it's saying is that you have a lot of N14 than N15.
No, N15.
Wait, N14.
Oh yeah.
Yeah, N14 than N15. Because
Yeah.
and the lighter one is going to go to the left, okay? And the heavier one is going to stay there.
Yeah, so it's just a different way that they showed it. You can centrifuge it and then see it in the test tube, or you can use a microscope, and then in the microscope, the darker part is more heavy and the lighter part is lighter. So it's just two different ways to show it, but you should see the same thing. So for Mosaic here, if I draw 1, because it's N15, it's going to be heavy, it's going to be at the bottom, and it's going to be all red. And then for the second cycle, we have equal red and equal blue. So we're going to have equal red and equal blue. And it's going to be a little higher because there's some N14 in it, so it's lighter than the first one. And then in the third one, there is mostly blue. But there's also a little bit of red. Oops.
To answer a student question, the question was, what was the biologist's goal for this experiment? So the whole point of this experiment is to determine the model of DNA replication. DNA replication, yeah. So it was proposed with 3 models: semi-conservative, conservative, and mosaic model. And after doing the experiment and seeing how the DNA replicate, they found out that DNA follows the semi-conservative model.
And if you have any last minute questions, just send them in right now. We have 2 minutes until we end.
Yep.
So some of the experiments that you have to know.
I'll put them in the chat.
Okay.
Yeah. So you can just say it.
You're welcome, guys.
So Griffith experiment, Avery experiment, Hershey Chase experiment, and finally Meselson
Stahl, and then the ricin ones as well. Ricin experiments.
If you guys don't know what ricin is, we remember you talk about the seed that like kills someone. Like, yeah, that's the experiment that we're talking about.
So I think ricin is actually something that's found inside that seed, which is poisonous because it stops translation. You can't make the proteins anymore. Yes, it's in castor beans.
All right.
You're welcome.
7:30. So we're going to be ending the session. Thank you guys for coming. This is our first session, so sorry for being a little bit
messy. We're
messy
still
and unorganized.
Yeah.
But we'll try to be better next time. So please feel free to come again.
Yeah, I hope it helped. And if you guys have any way you want us to improve or any feedback, you can email us, and we're definitely gonna like try to be better next time.